Okay. Well, it's my pleasure to introduce Abdel El Manira today for his presentation. And just a brief background for those of you who don't know Abdel. Before Abdel began to climb up the impact ladder in the world of spinal cord motor control, he spent his formative years in the early 1990s in Marseille, France, a graduate student, Francois Clairac, where he produced a lovely set of papers on presynaptic inhibition of sensory neuron terminals in the red swamp crayfish, Procambarus clarkii. At that time, there had not been many cellular physiology analyses of presynaptic regulation, though it's worth mentioning that some years prior to that, the same prepper launched the career of one Professor Calabrese. I first met Abdel during his graduate work as Melissa Coleman and I were also focused on presynaptic regulation, in our case, on the MCN1 axon terminals in the STG. After his graduate work, Abdel chose science over his climate preference and moved to Stockholm for his postdoc years in Sten Grillner's lab at the Karolinska Institute, studying the cellular basis of locomotion in the lamprey. This worked out so well for Abdel that he forgot to leave Stockholm. Instead, he married the boss's daughter, raised a lovely family, and eventually became faculty at the Karolinska, where he is now a professor of neuroscience. At some point in the early 2000s, Abdel shifted the lab away from lamprey and onto the zebrafish preparation to take advantage of the molecular genetic tools available there, while continuing to study the spinal control of locomotion, as you'll hear today. And he and his lab are continuing to publish a steady stream of lovely papers. And despite having left the crayfish behind more than 25 years ago, he remains appreciative to the value of small system circuit analysis, as is evident from his regular attendance at our monthly meetings. So without further blather from me, I cede the microphone to Abdel El Manira so we can learn what's happening in the world of smallish system spinal cord circuits. Take it away, Abdel.
I will share my screen now. And okay, you see my screen now?
Yep.
Okay, good. Well, thank you everybody for the invitation, and especially Nelly and Smita for the invitation to participate in this seminar series. And thank you very much, Mikey, for this generous and kind words of introduction. It is really a great pleasure for me to present our research at this SDG monthly meeting. The extended SDG community, or family as I would like to call it, means a lot to me. And I have been influenced and also taken inspiration from many studies on the SDG system. And among the first papers I was introduced to during my graduate studies are first from what I would call the influencers. And the first paper, among the first papers I've read is this paper by Ron Calabrese about presynaptic inhibition in the crayfish. And I really was inspired by this because it was the first time that I could see a really clear cellular analysis of presynaptic inhibition. I wasn't aware that presynaptic inhibition existed when I started as a PhD student. So I was lucky enough to find that or be able to record from primary afferents and also study presynaptic inhibition and try to emulate what Ron has already done. But Ron was very instrumental in many ways in my career because he joined our lab when we started developing the adult zebrafish preparation when he was in sabbatical, and he spent many months trying to develop the preparation, and he has helped a lot in getting to the point where we are today. And I really, many of our studies, he should also share the credit for them because he was really helping a lot at the beginning. The second paper I read is this one by Eve. And when I started again, I thought that there were excitatory and inhibitory transmitters. And by reading this paper, he just realized that a transmitter can be both excitatory or inhibitory, and depending on the receptors they activate and the ionic mechanisms the involved ionic mechanism. So this paper was really, I read it several times, and it was very helpful to me when designing my experiments I did on crayfish. I was also inspired by or introduced to neuromodulation by this review, or this influential review by Harris Warwick and Ron Harris Warwick and Eve. And I was also introduced to neuromodulation through studies in the stomatogastric. Moving on to my influencers, and I thought again that presynaptic inhibition, or the thought was that presynaptic inhibition was mainly restricted to primary afferents. And then there came this paper from Mikey Nussbaum, who showed that presynaptic inhibition also exists in modulatory neurons or descending neurons, projecting neurons. And later on, he showed how presynaptic inhibition can changes or switch between two modes of synaptic transmission, and how it configures circuit activity underlying motor behavior. And this is a study by Melissa Coleman and also our friend Pierre Meron and Mike Nussbaum. So this and many other studies in the SDG really have had a strong influence on me and shaped my way of thinking as a researcher. So it is a great pleasure for me to present our research, which is aimed at understanding how circuits in the brain and spinal cord generate and adapt locomotor movement. Locomotion can be expressed in different forms, and just showing here that there are different animals, mostly vertebrates here, that are expressing locomotion at different speed, and they can use either bipedal locomotion or quadrupedal locomotion, and they use different gaits to produce locomotion. Locomotion is intimately linked to many behaviors, from spatial navigation to social interaction. And while it is seemingly simple, the path to its execution is complex, with many levels of integration. And hence, it requires a higher degree of flexibility of its speed and vigor to align the motor action to the initial intention. So the increase in speed and vigor of locomotor movement is ultimately mediated by an orderly recruitment of motor neurons from slow to intermediate to fast. This orderly recruitment of motor neurons is driven by a circuit of interneurons called center pattern generator, and this center pattern generator has the ability to transform tonic command inputs from the brain into rhythmic and coordinated locomotor movements. So for this, the CPG must be endowed with mechanisms for precise control of the start, stop, and speed changes. It must be able to convert unpatterned motor commands into coordinated movement and with controlled speed and vigor. And finally, this CPG must integrate proprioceptive feedback to align the motor command to the actual motor outcome. And for us, as you know, the ability of the central nervous system to adapt locomotor movement to the behavioral context is ingrained in how the underlying circuits are assembled, connected, and recruited, and also modulated. And this is the focus of my presentation, which will be centered around the following question. First is, how does the CPG encode the change in the speed of locomotion? The second is how the CPG converts tonic and pattern command into coordinated locomotion. And finally, whether the adult vertebrates possess proprioceptors that monitor changes of the spinal cord tension. And for this, we used, to address this question, we use the adult zebrafish as a model system. And we have an ex vivo preparation with the brainstem attached to the spinal cord, and we leave a piece of muscle to be able to record the motor activity extracellularly, stimulate the descending commands extracellularly by stimulating electrode or optogenetically by expressing channel rhodopsin in specific descending neurons, and have access to spinal motor neurons and interneurons to be able to record their activity and also define their connectivity. So when we stimulate the descending command, we have a bout of locomotor activity, which can also be recorded in motor neurons and interneurons. And in extended trace here, you can see the motor output, the burst in motor output, and the phasic excitation followed by mid-cycle inhibition in this recorded intracellular, motor neuron. What we thought that there will be a ramp up of the activity or the speed of locomotion or the frequency of the motor activity, starting with slower frequencies and increasing to intermediate frequencies before reaching the final or the highest frequency and decaying to gradually decrease until it stops. And the first question that we have addressed is whether the locomotor CPG consists of a single or multiple circuits, and whether the locomotor CPG is distributing uniform excitation or input to the motor neurons, or whether there are dedicated pre-motor interneurons that are dedicated to slow, intermediate, and fast neurons. And for these studies, 3 persons were very instrumental. Is Jessica Osborne, who is now at Drexel University, Kostas Ambatsis, who was a postdoc, and now he's assistant professor at the Karolinska, and Jan Ransong, who was a graduate student, and he's now a professor in Shanghai. To map the connectivity between excitatory interneurons and the motor neurons, we use a transgenic line and focused on a specific class of excitatory interneurons called V2A interneurons. And we pre-labeled or back-labeled the motor neurons by injecting a fluorescent dye in the muscle to label the different motor neurons. And here you can see in red, the labeled motor neurons, and in green, the excitatory interneurons expressing GFP. And we did paired recordings by recording from a presynaptic excitatory interneurons, V2A interneuron, and postsynaptic motor neuron. And by stimulating a presynaptic interneurons, we have EPSPs in the motor neurons, and we map the connectivity in this way. We could also inject neurobiotin in the recorded neurons and reconstruct their morphology and also the site of their synaptic interactions. What we were able to show is that the locomotor CPG did not consist of a single unit, but it consists of 3 speed circuit modules with specific excitatory V2A interneurons that are dedicated to the slow motor neurons, and they are specifically connected to slow motor neurons. As you can see here, an action potential in one of these V2A interneurons elicits a monosynaptic EPSP in a slow motor neuron, and both the motor neuron and interneurons were recruited at the lowest locomotor frequencies that we could get in this preparation. Similarly, there were other excitatory interneurons that were dedicated or connected specifically to the intermediate motor neurons. And you can see again here the monosynaptic connection between the V2A interneuron and the motor neuron. And you can see that both neurons were activated at intermediate locomotor frequencies. And finally, we had another set of excitatory interneurons recruited at the highest frequencies, and they were connected to the fast motor neurons. And again, you have the monosynaptic connection and the recruitment of the two of the V2A interneurons and the motor neurons at the highest locomotor frequency. So This results, or by mapping the connectivity, we could show that the locomotor CPG, or the connection between the excitatory V2A interneurons and the motor neurons, they form three speed circuit modules. The next question that we have addressed is how does the spinal CPG convert tonic commands into coordinated rhythm? And for this, we focus on the connectivity between the V2A interneurons. And you can see here, there is a transgenic line again. This is a ventral side up of one segment of the spinal cord. You can see this excitatory interneurons on the left and right hand side. And this is a genetic line that was developed by a Japanese group led by Shinichi Hikashijima. So we did paired recordings from these excitatory interneurons, and we focused first on the slow V2A interneurons when we stimulate, and we recorded two of these slow interneurons. When we stimulate one of the interneurons, we had monosynaptic EPSPs in the other interneurons. Conversely, when we stimulated the other interneuron, we also had excitatory postsynaptic potentials in the first interneurons, as if the two interneurons are forming excitatory circuit with mitral excitation. Similar to the slow V2A interneurons, the intermediate V2A interneurons were also connected with mitral excitation, and the same for the fast V2A interneurons. So what we can say is that we have the V2A interneurons within each module form a recurrent excitatory circuit with strong and more frequent mitral connections. We then looked at the connectivity between the different circuits. And what we found is that the connectivity between the V2A interneurons of different modules showed a clear asymmetry in strength and probability in favor of connections from faster to slower modules. So the slower excitatory or V2A interneurons make connections with the intermediate ones, and the intermediate make connections with the fast one. However, the frequency of these connections or the probability of this connection is very low, and their amplitude, the amplitude of the EPSP is also very low compared to the connectivity from fast to intermediate or intermediate to slow, which had much higher EPSP amplitude in both cases and more frequent connectivity here. Then we looked at whether these V2A interneurons display any pacemaker properties. And what we found is that a subpopulation within each of these circuit modules, a subpopulation of V2A excitatory interneurons are endowed with pacemaker bursting properties whose prevalence and intrinsic bursting frequency relate to their modular affiliation and their recruitment speed. In other words, the slow V2A interneurons have the highest fraction of them, they display these pacemaker properties, while the fast module have only a small fraction of their this V2A interneurons displayed pacemaker properties, whereas the intermediate modules, we have 50-50, about 50-50% of the interneurons display pacemaker or non-pacemaker properties. And what we found is that when we looked at the size of the soma size of this, the excitatory interneurons displaying pacemaker properties versus those that are not displaying pacemaker properties, we found that those that are displaying pacemaker properties had a smaller soma size compared to those that do not display pacemaker properties. This allowed us at least to test the function of potential presumed pacemaker interneurons in behaving animal. The way we did it is by using 2-photon laser ablation in intact animals. And you can see here, we are scanning the spinal cord in an intact animal in control, and we focus on the interneurons that have the smallest soma size, and we target them to ablate them using 2-photon laser ablation, and we have spared the large interneurons that do not display pacemaker properties. And when we did this, so this is in control animal, you can see the animals that are swimming, and in the animals in which we have ablated the presumed interneurons with pacemaker properties, you can see that the animal, this is a live video, that the animals have difficulty to start locomotor activity, and they cannot move much in an efficient way. And we quantified this. So this is just the tracking the movement of the fish in control animals, while the one with ablated presumed pacemaker V2A interneurons, there was not so much movement and so much swimming. And here the quantification, this is showing bouts of locomotor activity in control in black. And for those in animals where we have ablated the pacemaker V2A interneurons, you can see that there is less locomotor activity or swimming activity that was displayed by these animals. In these animals, the escape behavior, which is not dependent on the activity of pacemaking V2A interneurons, and it is produced mostly by very fast set of V2A interneurons and the fast motor neurons, the escape behavior was not affected by ablation of these small V2A interneurons. So what we can conclude from here is that we have a 3-circuit modules that together with pacemaker properties encode the locomotor speed. And what we found is that we have this 3 modules of V2A interneurons with recurrent excitation and with the symmetrical connectivity across modules, which are targeting making specific connections with the different types of motor neurons. Each of these circuits, through these excitatory connections, can percolate the excitation to increase the speed up to a certain limit before propagating the activity to the next level and recruiting the next module to increase the speed even further, and so on to the fast one. And combined with the pacemaker properties, this circuit organization and pacemaker properties allow these excitatory interneurons to transform tonic input into rhythmic activity and increase the speed of locomotion. So what we have is a circuit that is able to produce ignition through activation of descending input and transform them into rhythmic activity, and acceleration up to a certain level before changing gears to activate the next circuit and the next circuit as to adapt the speed of locomotion to the behavioral context. So after defining this circuit organization and how it controls the speed of locomotion, we asked whether these 3 circuit modules are whether they are molecularly defined. And for this, what we have performed single cell RNA sequencing using transgenic lines, one to target the motor neurons, and we use this transgenic line in which GFP is driven by the transcription factor islet-1, or the for labeling for expressing GFP in the V2A interneurons, we use this transgenic line that we used before, in which GFP is driven by the transcription factor CHOX10. And what we have done is we've taken this spinal cores, we dissociate them, FACS sort them, did the RNA sequencing and the analysis. And these studies were done with a graduate student, Irina Palucchi, and the postdoc, Maria Bertuzzi. And we are in the process of analyzing this data, and I would like to share some of these results that have emerged so far from this analysis. So first, with the motor neurons, when we plotted the motor neurons, what we found that there were 4 clusters of motor neurons, 1, 2, 3, and the 4th cluster. And each of these clusters have a specific sets of genes that are differentially expressed For example, in C1, the cluster 1, the red one, has a specific genes that are differentially expressed in them. You have the cluster number 2, which also has some specific genes that are differentially expressed, but it also shares some of the genes expressed in the cluster number 1, but with a certain gradient. In addition, we have the third cluster that is different from these first two, and the fourth one that we think it's consists of motor neurons that are not fully differentiated. And I will explain this in details. So if we look more in details about what this 4 clusters express, if we look at the neuronal identity, and this is a marker of neuronal identity, it is expressed in all the 4 clusters. However, when we look at the motor neuron progenitor markers, we find that the 4th cluster still has some markers for cell differentiation, and we think that they are not fully developed because of this, because they are expressing markers for still cellular or neuronal differentiation. All the clusters, they have markers for motor neuron identity, and they also have markers for the cholinergic identity, although the 4th cluster has very little of them. Finally, we looked at the marker for axonal projection, or this is peripherin, which is expressed in the axons. You can see here that in the 4th clusters, this one doesn't seem to innervate the muscles, and it stays. It doesn't extend their axon to the periphery, whereas the 3 first clusters, they extend their axons to the periphery. When we look more closely to this, what we found that there are specific genes that are expressed in the 1st cluster, mostly this one of the NMDA receptor subtypes, and this estrogen receptor gamma, which is a transcription factor. So we use this to validate in which motor neurons these genes are expressed, and for this we used in situ hybridization using RNA-scope. And we labeled the motor neurons, and here we have labeled specifically the slow and intermediate motor neurons. And we could see that this NMDA receptor subtype is expressed specifically in these motor neurons. But it is not expressed in the fast motor neurons, again, which are, they do not express this NMDA receptor subtype. And we also analyzed the position of these motor neurons, and they are positioned in the part of the spinal cord, which is in which both slow and intermediate motor neurons are located. And we counted the number of puncta of this RNA-scope analysis, and we find most of the puncta are in the slow motor neurons, with and a decrease in the number of puncta in the intermediate motor neurons, while the fast motor neurons did not express much of this NMDA receptor subtype. Conversely, this 3rd cluster, which we think is a fast cluster, expresses a specific receptor for a nicotinic receptor subtype, and this nicotinic receptor subtype was absent in the slow intermediate motor neurons, and it was heavily expressed in the fast motor neurons. And again, you can see the position of these fast motor neurons is located in the dorsomedial part of the spinal cord, and the analysis or quantification shows a high expression of this nicotinic receptor subtype, which is not present in the slow or intermediate motor neurons. When it comes to V2A interneurons, we also found 4 clusters, and cluster 1, 2, and 3, and the 4th cluster, using the same reasoning as for the motor neurons. We think that these are not fully differentiated V2A interneurons, whereas the other ones are fully differentiated. And again, each of this cluster has a specific set of differentially expressed genes, and we have focused on 3 sets of genes that are differentially expressed in cluster 1, which is the estrogen receptor gamma. In cluster 2, SHOX2, which is a transcription factor. And in cluster 3, they have cholinergic markers. And we have transgenic lines that express GFP driven by this promoter for this transcription factor, or SHOX2, or we have also cholinergic GFP driven by markers for cholinergic transmission. So by using this transgenic lines, we were able to record from these interneurons, the V2A interneurons expressing estrogen receptor gamma, and we found that most of these interneurons, they were either firing in a pacemaker with a pacemaker-like activity or with tonic activity. And this is a signature of mostly slow V2A interneurons. The V2A interneurons that express SHOX2, the transcription factor SHOX2, they fire tonically with a certain delay or tonically with some spike frequency adaptation. And this is, again, it's a signature for interneurons belonging to the intermediate modules. Finally, the interneurons that are expressing or acetylcholine, they fired with a strong adaptation, or they are, all of them have either strong or moderate firing, adaptive firing. And this is, again, it's a signature for the fast V2A interneurons. So we have really the molecular markers that, or that we have clusters based on the genes that are expressed in motoneurons and interneurons that define the modular identity of the different types of neurons. Now, we also ask the question whether there are any specific genes that are shared between the motoneurons and interneurons that define the modular organization of these two types of neurons. And for this, what we did is we looked at mostly the genes that are specifically expressed in the slow and intermediate modules. And you can see here cluster 1 and 2 share many of these genes with a certain, but that are expressed in a gradual manner, and then that are different from the genes that are expressed in the fast module in the motoneurons. And all of these genes are also shared by the clusters of the slow and intermediate V2A interneurons, as well as, and those expressed in the fast motoneurons are also shared by the fast V2A interneurons. And when we looked at specifically to the shared transcription factors, again, we can see that there are the transcription factors expressed by slow and intermediate motoneurons are shared between this slow and intermediate V2A interneurons, and those expressed in fast V2A motoneurons are also expressed in the fast V2A interneurons. So we are still in the process of analyzing this and validating this results, but we have molecular data that shows that the V2A and V2A interneurons, as well as the motoneurons, are molecularly defined, and this molecular segregation of these different types of interneurons fits the physiological characterization that we have characterized before, and their connectivity and the modular organization. So what we have shown is that the locomotor CPG consists of 3 speed modules with specific connectivity with the motoneurons, and this modular organization endows the spinal cord with a gear shift mechanism to increase the speed of locomotion and adapt the locomotor output to the behavioral context. So I would like now to switch gears and talk a little bit about the importance of proprioceptive feedback and how it can control locomotor activity. As you all know, the classical view of proprioception is that we have, and in the control of motor activity, is that we have a centrally generated motor activity that is driving the activity of motor neurons producing muscle contraction, and the contraction of muscle is monitored by either 1A afferents or muscle spindle afferents, or 1B Golgi tendon organ, which are sent back through sensory neurons whose cell bodies are located in the dorsal root ganglia and make connection with the interneurons and motor neurons in the spinal cord, as well as sending axons to the brain. And the two core components of the motor of movement are considered as two functionally and anatomically separate entities: the motor command, which produces the movement, and you have the proprioceptive feedback, which arrives after a certain delay. However, it is the movement. When we move, we also change the tension that we apply on the spinal cord, as it is illustrated by this citation that states that the nervous system moves is beyond doubt. As the body moves, the nervous system has to accommodate and adapt to the movement it paradoxically produces. And we thought that whether there are any central proprioceptors which are located in the spinal cord, which will provide a direct feedback with a minimum delay to the central pattern generator, which will produce a sensory motor hybrid circuit that will integrate online the proprioceptive feedback. And there is, in the literature, there is such intraspinal proprioceptors or stretch receptor neurons, and those are in the lamprey, and they are called the edge cell, and they are located in the margin of the spinal cord, and they are sensitive to mechanical deformation of the spinal cord. However, it was thought that these are specific to the lamprey, which has a flexible body, and it does not exist in animals with a hard vertebrate column, or like in bony fish. So a study by this postdoc, Lawrence Picton, who started by looking at the neuroskeletal organization by using micro CT scan. So we have a fish in that we CT scan, and this is just a schematic reconstruction of such a CT scan. So you see the vertebra here, and then you have the intervertebral disc in blue and the spinal cord in white, with the ventral roots that are exiting the vertebral column. And when we look at the spinal cord, mostly at the level of this intervertebral disc, we see that there are enlargements of the spinal cord that we call dome. And you can see this in a raw data here. You can see this is one vertebra, and this is a second one, and the third one, and you can see the intervertebral disc here. When we see, this is a raw data at the level of the spinal cord, and you see here that there is this enlargement that we call dome, which is basically located at the intervertebral disc, where the maximum tension, where the maximum movement is applied on the vertebral column, and with the maximum tension applied to the spinal cord. We looked for neurons that could be located specifically in this region, and we found that there are specific inhibitory neurons expressing, using glycinergic neurons and expressing a specific transcription factor that are located in this, what we call domes here. And this is a ventral view of the spinal cord. And you can see here are 3 segments with these neurons located in the lateral margin of the spinal cord. So here are these interneurons, which are glycinergic and with GFP driven by this transcription factor. This dome highly expressed calbindin and also expressed OLIG2. And we think these are like some radial glia that form a scaffold that basically form this dome. The neurons, when we record from them, they have extensive dendrite, and with the processes that are extending in this dome, and they seem to be able to sense the tension applied to the spinal cord. These neurons are not found in the larva, are not found in the larva and not found in animals up to 2 weeks. They start being visible after 3 weeks of age of the animals, and they are found in the adult here. So in the larva, in 1 week old larva, there is no neurons in the ventral part. There are no neurons at 2 weeks old animals, and they start being visible only after 3 weeks. When we look more closely at the expression of DBX, which is a transcription factor expressed by these neurons, there is an increase of expression of DBX up to 2 weeks, and then it decays after 3 weeks. If we increase the contrast between 2 and 3 weeks, what we see is that there are some cells that seems to be migrating from the central canal to settle in the dorsal part, and this could be this lateral neurons located in the ventral part of the spinal cord. To study this more closely, we used again single cell RNA sequencing, and we dissociated neurons from transgenic line in which we express GFP in a Cre-dependent manner in neurons that express DBX1. And we fact sorted the neurons and did the RNA sequencing and the analysis. What we found is that we have 2 neuronal clusters and 1 and 2 and 4 glial clusters that we have excluded. These neuronal clusters, they express neuronal markers, which are not expressed in the glial clusters. They express markers for glycinergic or inhibitory synaptic transmission and markers for synaptic formation. To determine which of these clusters correspond to the lateral neurons, and just to remind you, what we have done is to record from the neurons and use what we call patch sequencing. And by recording the neuron, we can basically pick the neurons that we have recorded from, and we picked from the lateral neurons and from the interneuron population, which is found in normally as intermingled with the other neurons. And this interneuronal population is belonging to the V0D population, which are inhibitory commissural interneurons. And by picking these neurons and doing single cell RNA sequencing, we could just determine how many of the genes that are expressed in the lateral neurons, whether they are shared with the cluster number 1 or cluster number 2. And we found that the majority of the genes expressed by the lateral neurons are shared with the cluster number 1, whereas the majority of the genes that are expressed by the V0D interneurons are shared with the cluster number 2. And here just see the highly expressed gene in the lateral neurons that are not expressed in the interneurons. And among these genes are those that are encoding for the mechanosensitivity, especially piezo2 channels. To validate the expression of piezo2 channels in the lateral neurons, we used RNA-scope. And here you can see the GFP expressed in the lateral neurons, and these are the interneurons. And you can see that the piezo2 RNA-scope is limited to the lateral neurons, which is highly and specifically expressed in them, and it's not expressed in the interneurons, and then just showing this. And we have also done quantification for this, and I will not show you this. To determine whether these neurons that have piezo2 channels, whether they are also activated by mechanical deformation of their membrane, we use the mechanoclamp recorded or patched from a neuron, these lateral neurons, and have a glass pipette that is, or mechanical probe that we can move very quickly to activate or make a deformation. As you can see, here is GFP in this lateral neuron. This is the electrode before and after poking the neuron. And by applying this mechanical activation, we see that we have an inward current with a fast activation, a fast inactivation, which is characteristic of piezo2 channels. And this current is blocked by ruthenium red, which is, among others, blocks piezo2 channels. And you can see that there is an increase in the amplitude of the current as a function of the amplitude of the stimulation. These mechanosensitive lateral neurons, they are sending their axon, they have extended dendrites that are going into the domes where the maximum tension is applied during movement on the spinal cord, and their axon is projecting to the other side of the spinal cord and projecting rostrally. And on the way to the brain, it makes contact with the corresponding neurons on the other side. As you can see here, these are other V2A, or not V2A, but the lateral proprioceptive neurons on the contralateral side that are connected with the axon of the neuron from the opposite side here, and they are making connection with them. And we confirmed that these connections are mediated by mediating synaptic interaction by recording from a caudal neuron and on one side and the rostral neuron on the other side. Stimulation of the caudal neuron produces an IPSP in the rostral neuron, while the reverse is not valid. There is no IPSP produced by stimulation of the rostral neuron in the caudal one, just because their axons are projecting only rostrally. These neurons, they use GABA and glycine, and we can block this IPSP by blocking GABA and glycine receptors. We could also use mechanical activation of the neurons by just bending the spinal cord to one side or the other and recording from a neuron. When we record from one of the neurons, if we bend the spinal cord on the opposite side, we have a large IPSPs occurring that are mediated by the bending of the spinal cord. But if we bend the spinal cord on the same side as the recorded neurons, we see no IPSPs and we don't see any EPSPs either. And these IPSPs are sufficient to block the firing of these proprioceptive neurons on the opposite side and inhibit them efficiently, while the bending on the same side does not affect the firing of these proprioceptors. We made sure that the bending is produced by these lateral proprioceptive neurons in the spinal cord. As you can see here in control, we have IPSPs or inhibition mediated by the bending of the spinal cord. And when we ablate these proprioceptive neurons on one side, the same bending of the spinal cord does not produce any IPSPs. So having determined that there is a movement-coupled feedback, inhibitory feedback mediated by intraspinal proprioceptors, we wanted to know which are the types of neurons that are targeted by this proprioceptive feedback. So what we have done is we recorded from CPG neurons, motor neurons, V2A interneurons, and V0D interneurons, and penned the spinal cord on the opposite side or on the same side. And here just show the analysis of this. So this is a recording from one motor neuron, and each line is showing one motor neuron, and the EPSP amplitude or the number of the EPSP amplitude is shown in a color code. And you can see that there was no IPSPs induced either by the bending on the opposite side or on the same side in motor neurons. There was a strong inhibition occurring in the V2A interneurons, which are the rhythm-generating interneurons in the spinal cord. You can see this is a strong inhibition produced by bending of the spinal cord on the opposite side, while there was no inhibition induced by bending of the spinal cord on the same side. And finally, the V2A interneurons, V0D interneurons, which are the ones that are mediating left-right alternation, were not affected by the movement coupled feedback produced by these proprioceptors. So it seems that these proprioceptors are targeting specifically the rhythm-generating V2A interneurons to affect the locomotor CPG. And when we record from these V2A interneurons and depolarize them to make them fire in the absence of locomotion, we can make rhythmicity just in these neurons by applying movement to the spinal cord on the opposite side. So what we can say is that we have this rhythm generating, producing the locomotor command that is propagated to the motor neurons to produce a contraction of the muscle on the one side, and on the opposite side, the proprioceptive feedback or the proprioceptive neurons will be activated that will target the V2A interneurons to terminate their activity and produce the phase transition to alternate between the left and right side. So having determined this, or characterized this intraspinal proprioceptors and their connectivity with the, specifically with the V2A interneurons, wanted to know how they contribute to normal locomotion. And again, for this, what we used is specific ablation of this intraspinal proprioceptors and tested behaviorally in freely swimming animals. So here in control animal, you can see that we can induce fast locomotion, and you can see we are looking at the movement of the head and the tail. The movement of the head is in red, and the movement of the tail is in green. In the animals with ablated proprioceptors, they have high difficulty starting locomotion, and even when they start locomotion, they are moving less efficiently and with very large movement produced along the whole body, both in the head and in the tail. And this is the quantification here. So we have this is the curvature, and this is the head, and this is the tail. In control animals, it's mostly the tail that is moving in an efficient manner. This is time, and you have movement of the tail very fast to produce efficient locomotion. In the animals that have with ablated proprioceptor, intraspinal proprioceptors, there is movement along the whole body and with large curvature, but with less efficient locomotion because they don't move very fast and they cannot increase their speed very fast. So just to summarize what I have shown, so in the spinal cord or in zebrafish, we have intraspinal proprioceptors located at the intervertebrate joint that is sensing the tension that is applied to the spinal cord. They are specifically connected to the rhythm generating V2A interneurons and produce the phase transition and pattern or time the activity between the left and right side. So by starting from this view of proprioception with only peripheral proprioception, producing with the two entities for movement control, the motor command and the proprioception acting as two separate entities with the feedback coming with a certain delay, our results show that there is internal proprioceptors that act both as sensory neurons sensing the bending of the spinal cord, as also as interneurons to time the activity of the rhythm generating interneurons and form a hybrid sensory motor circuit for efficient locomotor activity. So we have really our analysis has been inspired a lot from what the STG community has been doing, just really targeting and trying to understand the single cell, their connectivity and relation to behavior. And we have found that we have like a modular organization of the locomotor CPG and also this hybrid sensory motor organization that exists between and with the role of proprioception in timing the activity of the locomotor movement. And we are analyzing this more in details by looking at how this proprioception and the inhibition from these proprioceptors really times the activity even in intact animal. And I just show some analogy between the locomotor circuits in vertebrates and the somatogastric system in crustacean. So this is from Cajal, a drawing from Cajal. We have what I would call the STG ganglion with the proprioception. You have also proprioceptors here, and you have the descending command from the brain that produce the initiation and also regulation of this locomotor CPG. And you have many in the stomatogastric system. We haven't started looking at neuromodulation, but I hope we will be able to do that in the future. So to finish, I would just want to thank many people. And first is Ron. I really cannot thank you enough for all your contribution. And I think you have a large credit to for the studies that we have done, because you really you were instrumental in developing the preparation, and I'm very thankful for your support and friendship, Ron. Thank you very much. The second is Eve. Eve, you are an inspiration, both for as a scientist, as a leader. I really appreciate discussing with you, and also having input from you, and also reading your book. It was a joy reading it. And also Ron Harris' work, for his wisdom and support while he was in Stockholm during sabbatical with Oli Keen. We had many discussions with him. And I must also say that also have a lot of inspiration from Ad Silverstone, and also Maurice Moulin, the late Maurice Moulin, and all the group from Arcachon that are very good friends. Last but not least is Mikey. You are my best friend. I thank you for your generosity, insights, support, and also for telling me all the papers that I have forgotten to mention, or the studies that were done before, and to remind me that science is incremental, and it is always built up, knowledge built up on old knowledge. So thank you for that. And thank you for always being happy together here with our friend Ansgar Bushges, and also for always arranging this mini dinners, our steak dinners, as we call them, here with Ron and Farzan and Mikey, and with many others like Don and Jorge and Patsy Dickinson. So I really am grateful for being able to interact with this community and learning a lot from you. And I thank you for the opportunity to present our results. So I just want to finish. I will not spend a lot of time to thank people in the lab who have been very, it's again trying to build this family that as you have in the stomatogastric system. And I have been also brought up as a graduate student in France in a really close interaction and very friendly environment. Many former postdocs and students, and also again, Ron and Ansgar for the collaborations, Claire, Herwig Bayer, Joe Fecho, and Shinichi Kachijima for providing the many different transgenic lines that we use, and the funders that will support our studies. Thank you very much, and I would be happy to answer questions. And probably I stop sharing.
Yep. Lovely talk, Abdel, lovely talk. I have several questions, but I will wait and see if there are other questions first. Ron?
So I have sort of two questions. One's more conceptual and one's more technical. So the conceptual question is, do the intraspinal proprioceptors, do they have module-specific connections? And the technical question is, your fourth cluster of motor neurons, could you test that by labeling motor neurons directly and sorting? You wouldn't expect cluster 4 if you back-labeled motor neurons.
Yes, when we back-label motor neurons, we see some in the line, we have some GFP neurons that are not projecting outside by backlabeling the motor neurons. So there are like dormant motor neurons that are like a reserve pool that is not projecting. And it is known that when there is a injury to the spinal cord, there can be some regeneration of motor neurons and others that start projecting outside the spinal cord. But that's what we think, that they act like as a reserve pool, but we don't know whether they are just there and waiting, or they are just responding to injury or something else. So those are the experiments we would like to do. Okay.
And the module specific connections?
Yes. So this is what we are doing. The proprioceptors are mostly connected to the slow and intermediate V2A interneurons, not to the fast ones. Especially, they do not connect to the V2A interneurons involved in escape. Escape is very fast movement, and probably the proprioceptive feedback will not have enough time to adjust that or regulate it. So we have module-specific connectivity, mostly to the intermediate and the slow modules, and not to the fast one.
I have a follow-up question, actually, to Ron's question, based on your answer, because I was struck by one of the early slides you showed, where the fast V2A neurons very few were intrinsically rhythmic. They mostly fired tonically. And so I was wondering how you got rhythm out of tonic activity. And so then I was assuming was the result of this feedback from the stretch receptors, but apparently they're not. So how do you have a rhythm generator without a rhythm?
But there are other commissure neurons, interneurons, that produce the inhibition.
So, right. So I secretly knew that, but I was really curious as to why you don't include that group of neurons as part of your so-called rhythm generator if they're necessary to generate the rhythm?
Because we are still working on them, and we don't know enough about their connectivity. So it's work in progress.
Got it.
Okay.
Beautiful talk, Abdel.
Thank you.
I was wondering about the integration of the central and peripheral proprioception. Are they targeting the same central neurons? And do you know anything about how they might control the rhythm the same or differently?
Central and peripheral. We don't know what type of peripheral proprioception there is in zebrafish. So they don't have muscle spindle afferents. And so far they have skin receptors, but we don't know how they are connected to the interneurons and the CPG. So that's we don't know a lot about. But the central proprioception, I think the sensory part is very important, or at least the results we have now, it's very important in timing the activity in the intact animal. And I think just to relate to Mikey's results, I think the inhibitory interneurons, they just give like a broad band or like a range of when the activity will alternate, but the proprioceptors really time that activity more precisely than the central inhibition in the intact animal.
Thanks.
Did I understand you to say that there are no muscle proprioceptors at all?
No.
There aren't any.
No.
That's so odd, because muscle proprioceptors go back to, crayfish and lobsters have stretch receptors out there in the muscle, and
Yes, I think.
So-
Yeah, go ahead.
so you're sure that there are none, or just so there's
I am sure that no one has found them yet.
but
And Melina
So
Hale.
where did, I mean, when, I mean, isn't it odd to imagine that you would have mussels with no proprioceptive, and then in larger fish and big other fish, there are none there either? Or what about in lamprey?
The lampreys, they don't have muscle spindle
So when did
afferents.
they come?
It's a good question. I don't know if other fish have them, but I know, like, for example, Melina Hale in Chicago, she thinks that there are some the skin receptors that might act as sensing the tension applied on the body
Oh,
through
so they're sort of
yes.
stretch.
Yes.
Weird.
Yeah. But they have the
Nope.
simple
Did
ones.
anyone else know this?
Nope.
Yeah. I mean, it's a well-published thing that muscle spindles only appear in tetrapods, and amphibians are the earliest ones. And I think there are some sort of similar things in fishes in some places, in jaw openers and things that people have found, but generally fish don't have muscle spindles.
But the jaw muscles
Well,
in fish do?
I think there's some, I just Google it. It's you can.
When I first learned
There's
this,
some, like, weird-
I
I mean,
looked
generally
and
they
looked and looked and tried to find some reference for muscle spindles in fish, and I saw just some weird anatomical paper that there might be in the jaw.
Yeah.
Might be.
So where do they first show up in frogs or?
Yeah, frogs.
So then the crayfish stretch receptors, the lobster stretch receptors, the classic, basic and tonic ones, they go away at some point? The MROs?
Those are really different. They're really very different kinds of beasts. They're not in muscle. The MROs are span a joint of the abdomen, and they're in these special sort of fibers that are not contractile.
So the chordotonal organs, they are just arranged in parallel to the muscles, but they are not contracting themselves. They don't have muscle part.
Well, I really just learned something, so thank you.
Wow, good.
But obviously Dirk and Ron knew it. So,
But it's
Yeah,
also, they
well-
haven't gone away. I mean, the question is what kind of proprioceptors and muscles the last common ancestors of arthropods and vertebrates had. I don't know if that's known.
Yeah, I think that.
I mean, MROs are, for all I know, are an arthropod invention, right?
The chordotonal organs I think of is more like Golgi tendon organs,
Yeah.
Yeah.
more analogous to them.
Abdel, I have a different question. In one of your early schematics, you were showing the location of the different modules of motor neurons, and it appeared that none of those modules were located in the ventral section of your spinal cord drawing. Are there other neurons that live there, or is that just a cartoon that was not effectively showing locations?
Which one of the, I think that in the ventral part, whether they are so what is your question is whether there are neurons in the ventral part of the spinal cord?
Yeah, well, I thought the ventrolateral region of the spinal cord was occupied by motor neuron usually, but you showed us the location of your 3 modules of motor neurons, but none of them were in the bottom third or so of your drawing of the spinal cord. So is that just a drawing thing, or,
Yeah,
is there other neurons?
No, it's just a drawing. The slow motor neurons in the ventrolateral part, the intermediate in the mediolateral,
Right.
ventromedial part, and then the fast ones in the dorsomedial part.
Okay, so it's just a cartoon.
Yes. And the proprioceptors are in the white matter, outside the gray matter of the, yeah.
Also, did you, when you were doing your analysis of expression in the various motor neurons, did you find any genes for co-transmitters?
For co-transmitters,
Yeah.
yes. Yes, they express, some of them express glutamatergic, or they have genes for glutamatergic transporters, vesicular glutamatergic transporters.
Uh-huh.
However, when we do paired recordings, they don't release glutamate.
So might they be using glutamate at the muscle?
Possibly, or to other, probably to other unknown targets.
Yeah.
At least when we looked at them to the motor neurons.
And
They
were they
don't use-
module specific, or all the modules were expressing?
Then so let me rephrase. In the motor neurons, we didn't test. In the V2A interneurons, as I showed, some of them, they have also cholinergic markers.
Uh-huh.
Okay. And
Okay.
those V2A interneurons, although they have cholinergic markers, they don't release acetylcholine on the motor neurons. They use glutamate.
Okay.
The motor neurons, they have VGLU2 or transporters for glutamate, vesicular glutamate transporters, but we haven't tested whether they release glutamate.
Abdel, I have a question. What proportion of neurons in the spinal cord are uniquely re-identifiable as stomatogastric neurons are?
They are uniquely identifiable as groups, most of them. But then we have certain neurons that are found 1 or 2 per segment, hemisegment, and they can be uniquely identified. And we could trace their connectivity to motor neurons very reliably from one preparation to the other. So it's not 1 per spinal cord, but 1 or 2 per segment, hemisegment.
How about in terms of the genes that are expressed in different neurons?
Well, for the genes, we didn't look at that because it's really time consuming. We have to pick them one by one. And we need to have like 300 or 500 to be able to sequence them to have a meaningful analysis. But we found, for example, a specific type of V2A interneurons that use, instead of using glutamate, they use acetylcholine as transmitter. And those are very, playing a major role for defining during escape. And they are found 1 per hemisegment. And those also, we have done also RNA sequencing from them, but we didn't look at deep in the sequencing. We didn't have enough depth to be able to compare them to the rest of the V2A interneurons. So the answer is that there are some single neurons that we can identify. There are few of 1 or 2 per hemisegment with specific connectivity and specific function. The ones that I have talked about today, we cannot uniquely identify them individually. We can identify them as populations or subpopulations.
Have you done any transcriptomics on the reticulospinal neurons?
Not yet. Not yet, no.
Abdel, I have a question. I may have missed it, but how does a fish begin to swim slowly and not accelerate? Because if these modules presumably all receive some sort of command neuron, like, for example, the equivalent to the mesencephalic locomotor region in mammals. They would all receive the input unless they have different excitability, and that is recruited in a graded way from slow to fast, for example, because I'm not sure if I understand whether you can select the slow rhythm without accelerating almost automatically, giving your little loopy kind of connection between these pacemakers.
If I have to ask, can I share again my screen and show you one more slide?
Of course, yeah. Go ahead.
Okay. Just so we are looking at, very briefly, we're looking at one of the descending inputs. So these are the descending input from the brain. So back labeled neurons. This is the hindbrain. This is the midbrain. And we looked at this nucleus called the nucleus of the medial longitudinal fascicle. And there are neurons, there are some neurons that are medial and some that are lateral. The lateral ones express VGLUT1, the medial one express VGLUT2. When we did calcium imaging and induced low vigor swimming, or this is head fixed animals, and this is spontaneous swimming, with low vigor swimming with slow frequencies, high vigor with high frequency. So when we do calcium imaging, the median neurons expressing VGLUT2 are active bilaterally. This is left, this is right, active bilaterally during low vigor swimming. The VGLUT1, the lateral neurons, are not active during what we call low vigor explorative swimming. But they are active during high vigor swimming. So there are different descending neurons that are activated either during low vigor or high vigor. The low vigor neurons still maintain their activity during high vigor swimming and fast swimming. But in addition, we have this, the other population. So, and we don't understand this fully, and we don't know how they are connected to the modules, but we think that there is also modular organization of the command descending from the brain. I don't know if this answers.
Yeah, maybe, but I thought specifically that another way to do this is that the interneurons in the spinal cord had the different modules might have different excitabilities.
Yeah, they have.
They do. Oh, I see.
Yeah. The slow I have the lowest or the highest excitability, and then it's graded.
Okay. Oh, so I missed that.
It is very hard to recruit the faster one.
Yeah.
And it's very easy to recruit the slower one. In addition, the input is also modular from the brain.
Okay.
So one final question. Ramping up from zebrafish up the phylogenetic tree, is there enough known in any other limbed vertebrate, maybe amphibia, to know whether there are comparable modules for locomotion there?
Amphibian, I don't know if anything is known more than in the tadpole.
Yeah, okay, the tadpole.
About the circuit, but the tadpole, maybe there are some modularity, but I don't know. I don't think it's known about the connectivity there enough about the different types of motor neurons, slow versus fast and so on. But in the mouse, I think based on data from transcriptomic data, there are subpopulations of in the different types of interneurons and motoneurons. Of course, they have slow, fast, and fast fatigable, fast resistance, and slow resistant motoneurons. And then in the interneurons, they have different subpopulations, and it's not known about the connectivity, but it's known that there are subpopulations with similar subdivisions, at least molecularly. But we are, I don't think there is enough known, at least in an adult system, about the connectivity of the network to be able to make enough the sufficient comparison. But hopefully the transcriptomic data can be a way to guide us towards having some answers there.
Can I piggyback on that? So forget about limbed vertebrates, but even within teleos, there's pretty radical differences in how they actually produce locomotion, right? So anything from undulating swimming to basically I think tuna just basically use their tail fin to swim. And I mean, it's an interesting question that, assuming you don't have an answer to, but like the modules that you describe could be co-opted for all of these different types of coordination. It's just a matter of left, right, and intersegmental connectivity potentially. Right?
But I don't think for the limb animals, because you have to define, because you have multiple joints and you have to coordinate across different joints. I think the left, right is very easy to intuitively understand. I think when you have multiple joints and the coordination has to be how to say, precisely controlled.
The principles of sort of ignition and acceleration and the things that you described could still apply to whatever the coordination is.
Yes.
Yeah,
In the broad parts, yes.
The other thing you're not considering is that the gear shifts in limb vertebrates often involve gait changes.
Right.
And the modules could be different among the gaits.
Yes.
But I think that may be true for fish too, to some degree, like from undulation to, getting the power stroke coming from the tail fin and all of these kinds of things. And I mean, fish do have fins that are in many cases not just purely used for stabilization, right? So
the pectoral fin could be informative. The circuit of controlling the pectoral fin movement could be informative, but we haven't done any work on them yet. But there are also changes, developmental changes. So we see like some in the larva, some of the neurons, like some fast neurons, are also involved in swimming in the larva. Whereas those neurons in the adult, they become exclusively dedicated to the escape system, but not to the swimming system.
Fish have another really crazy problem in that their center of buoyancy is below their center of mass, so that they're statically unstable and they constantly have to be using bilaterally paired fins to stay dorsal side up. When you kill them, they go belly up.
Is this the end? The last word?
So belly up is a good place to end.
Oh, that sounds like a note on which to quit.
Yeah.
Thanks so much,
Abdel.
Jeff.
That was a
Bye, guys.
wonderful
Thank you all.
Great
Thank you.
Thank you, Abdel.
Yeah.
Great talk.
Nice evening. Yeah, thanks.